In this video series for the urinary system, we're going to be looking at the kidney's anatomy. So hopefully you'll view this before lab. The kidney's vasculature, which is very important. We'll take a look at the anatomy of the ureters and the bladder, and then at the end we'll examine the micturition reflex and how the autonomic nervous system and the somatic nervous system play a role. So as we get started, as we view the urinary system, we can see that there are 6 organs that are going to be considered. So there are 2 kidneys, we have 2 ureters, we have the urinary bladder, and then 1 urethra. So just by looking at a picture of the abdominal cavity, we can start to pick up something interesting by how the kidneys are placed. So the right and left kidney are not symmetrical. The left kidney is higher, the right kidney sits lower. And the reason is because above the right kidney, remember, we're going to have a very large liver that takes up a lot of space. So this right kidney is lower to be able to compensate for that. So an actual human kidney, it's about the size of a bar of soap. You can see it's about 4 inches in length. Each kidney has a indentation on the medial side, and we call that the hilum. The hilum is where we're going to have our vasculature, our ureters, our lymphatic vessels, nerves come into the kidney. And you're going to see a hilum on many different types of organs. It's normally a medial indentation on the organ. We know that the ureters then are going to drain away urine from the kidneys. And when we look at the kidneys and the ureters, they are positioned what we call in a retroperitoneal fashion. So the peritoneum is the membrane covering in the abdominal cavity, and these kidneys and ureters sit behind that. So if you remember in some of your rat specimens, the kidneys were actually behind a membrane. They were shiny and they were covered up by that transparent membrane because they are retroperitoneal. So the location of the kidneys, they are sitting on either side of the vertebral column, and they have protection on 3 sides. So in front of them sit the abdominal organs, behind them sit the musculature and part of the skeletal system, so the ribs are there as well. If we take a look at their positioning, we can see a couple things. So in front, we can see are the abdominal organs, and it's helping for protection. We also have them sitting behind that membrane. So you can see in here we're going to be behind, here's another, this is our peritoneum, we're behind that with the kidneys. As well, you can see here's the vertebrae, and as we wrap around here, we're going to have the ribs. We also have musculature, it's part of the abdominal wall and our back muscles that are going to be protecting the kidney. Around the kidney, you can see all of that yellow. That's all fat and adipose tissue, and that fat is actually helping to anchor the kidney. It's sort of standing up. If you look, they're just hanging out, and gravity is not pulling them down because they are embedded in this adipose tissue, which helps to keep them in position. We also are going to be looking at, and there's layers to the kidney, and this fat layer is actually one of the layers as well. Something that is interesting to think about as you consider the fat that is helping to hold the kidneys in place is something called renal pitosis. So if that fat shrinks or decreases, for instance from weight loss or extreme starvation, especially if it happens quickly, the kidneys can fall, and when they fall, the ureters kink. That causes urine to back up in the kidney, and now that we've talked about the glomerular filtration rate and how it works, if we were to increase the amount of fluid that is in the kidney, eventually it's going to increase in the nephron and then the glomerular capsule, and so we would disrupt our net filtration rate. The capsular hydrostatic pressure would increase, and that would decrease our glomerular filtration rate. So if we look then at the connective tissues that we have that build the wall to our kidney, there are 3 layers. So right on the surface of the kidney is the fibrous capsule. That's the deepest. It's collagen that sits right on the surface of our organ. Surrounding the kidney then is what we call the perinephric fat capsule. So it's right, that yellow adipose tissue right around the kidney. And then we have the renal fascia, and that's this most superficial layer. Fascias are connective tissues, and in particular this is going to help to hold the kidney in place to the body's wall. So if we come back, we can see right on the surface of the kidney we have our fibrous capsule. So you cannot separate it from the kidney. It's collagen that's going to be making that capsule. Around that we have our adipose tissue that's helping to anchor this kidney in place. And then around that we have the renal fascia, which is helping to anchor the kidney to the body wall. So we're going to look at the major structural landmarks in the kidney. And we're going to go through these, and you will be able to pause on this slide to write down your vocabulary words. Before we got started though, I wanted to let you know that sometimes people use this renal sinus. I would write down also renal pelvis. It's a similar type of structure. And we're going to go through these on a picture of a kidney. So to get started with the anatomy, you can see the kidneys look brown when you look at them, the outside covering. And remember, it's a highly vascularized organ, and so you're going to have a dark color to organs with a lot of blood vessels in them. Right on the surface, we have the fibrous capsule. And then as we come in, we see that the kidney has different areas to it. And so we have this area with all these triangles or pyramid-shaped things, and then on top of them is an area with all these stripes. And so to begin with, we have 2 major regions. We have the cortex. Remember, cortex always means outer, so it's this nice rim that's around those pyramids. And then we have these triangular-shaped things on the inside. And so where we have these triangular or pyramid shapes, this inwards is considered the medulla, medulla for middle. In this kidney, right, the medial side here, we have this indentation right here. So that's the hilum, and you can see the ureters are entering here. This picture has the blood vessels removed, but the blood vessels and lymphatic vessels would be entering the kidney right here in this hilum region. So let's talk about these pyramids. These are called the renal pyramids. And so the renal pyramids are striated. They have little lines in them. And those little lines are actually the connecting ducts. At the bottom of each pyramid are holes. And those holes are called, the holes are the actual openings to the collecting ducts. And where those holes are, we call that the papilla. They're in this bottom, the point to the pyramid. In between the pyramids, we have what we call renal columns. And so inside these columns, you can see there's blue lines and red lines. That's the vasculature. So arteries and veins are going to be coming up through those columns. When we have a pyramid and the associated cortex above it, we call that a lobe, so a kidney lobe. So in this picture of the kidney, we have 1, 2, 3, 4, 5, 6, 7 lobes. The renal pelvis is this area where we have excuse me, where we have drainage right before we go into the ureter. So the pelvis is part of the drainage system. The pelvis is sitting in an area that we call the renal sinus. So underneath the medulla, we have a space. Think of it like as a hollow hole where the ureter is going to be coming in, and where we have the vasculature coming in. So a sinus means a space. As we look at how we collect the urine, we have different structures associated with it. We have the minor calyx or calyces. They are going to collect urine from one kidney lobe. We have a major calyx or major calyces. They collect the urine from the minor calyxes. And then the pelvis is going to collect urine from all of the major calyces and pull it towards the ureter. So if we were to look back at our picture, here's our renal papilla, the point of the pyramid, and our collecting ducts that will be collecting the urine as it is coming out of the nephrons. All of those collecting ducts start to drop that urine into a cup that is surrounding the bottom of that pyramid. And so the urine is being caught in that cup, and that cup is a minor calyces. As you have minor calyces merge together, the merging point is called the major calyces. So for instance, right here is a major calyce, and it's coming from 1, 2, 3, and this hole is going back to another kidney lobe. So that would be four minor calyces coming together to make this major one. Here is one minor, here's a second minor, and where it's coming together would be a major. Here's a minor, and where they merge, that's the major. Where all of those major calyces merge, that's the renal pelvis. Here you can see it's on a picture of a real kidney. So you can see when you have a specimen and you cut it, the actual structures are not as clear cut as they are in the pictures, but we can see here is a minor calyce, and where they're coming together from other pyramids we have a major, and those major calyces as they merge are going to become the renal pelvis. So as we look at the microscopic anatomy, we have looked at the nephron already and the components to it. But if we consider nephrons, we actually have two types of nephrons in a kidney. And the types are based on a little bit of their anatomy and then their placement in the kidney. So cortical nephrons are in the cortex, and that's where the majority of neurons lie. They have and they cover most of the regulatory function on the kidney because they are 85% of all nephrons. A small number of nephrons are going to sit in the area that we call the juxtamedullary area. And the juxtamedullary area means right next to the medulla. They have a very long nephron loop or loop of Henle that extends into the medulla, and that is going to give these neurons a function of creating very concentrated urine. And we're going to be looking at that physiology in lecture. So if we take a look, the corticonephrons sit way up in the cortex. Only part of their nephron loop is even in the renal medulla. However, these juxtamedullary nephrons, they sit right next to the medulla, and their loop of nephron is going to be coming way down into the renal medulla. And what's significant about this is the renal medulla has a salt gradient in it, and that salt gradient is going to work via osmosis, and it's going to help us to concentrate urine. You can see the collecting ducts for these nephrons, all of them collecting ducts come through the medulla, and here at the papilla you can see are the openings to those collecting ducts. They open and we catch that urine in a minor calyx.